This video will be on chapter 15. It'll be the first part of the genetic code and translation. We're going to look here at a little bit of background on protein structure and also looking at some of the tools that are used in translation, the transfer RNAs, which actually do the translation between a nucleotide code and an amino acid code. So proteins are made of amino acids, and all amino acids have the same basic structure. There's an N followed by two C's. The second C is actually a carboxyl group. The middle C has what's depicted as an R here, and this is the side group. This is the place where different amino acids actually differ in their structure. When proteins are made, we line up amino acids and link them together. And here is the first amino acid. This will always link up in the same order, so NCC, and the carboxyl group of that first one will link up with the amino group of the next amino acid. In the process, you lose a water molecule and you form a peptide bond. Now, it's important to note that proteins are polarized molecules, just like DNA and RNA were polarized molecules. So because these always line up in the same manner, we'll end up with proteins that have one end being the amino end, or the N-terminus, and the other end being the carboxyl end, or we also call it the C-terminus. Now, there are 20 amino acids that are commonly used in proteins, and these are shown here. I don't expect you to know the specific side chains of all of these amino acids, but I do expect you to know how they are characterized and just the general amino acid structure. So these can be grouped into hydrophobic side chain amino acids in the first group here, hydrophilic or polar side chains, and then acidic or basic side chains. So the nucleotide code specifies amino acids. So in 1953, Watson and Crick published the structure of DNA, and at that point, people really began to accept that DNA is definitely the genetic material, and soon after that, people began to ask, how is it that that nucleotide code specifies the production of proteins? And people started to consider different possibilities at that time. One possibility people considered was that maybe two nucleotides could specify one amino acid, but that was quickly ruled out because actually two nucleotides only give 16 possible combinations with those four bases, and so that is actually not enough combinations since there's 20 amino acids. People looked at some other possibilities, 3 or 4, and 3 nucleotides gives 64 possible combinations. People thought, well, that's really too many since there's only 20 amino acids, but nevertheless, these guys, Francis Crick, so this was the same person who did the structure of DNA, and another person, Sydney Brenner, did some very elegant experiments that were genetic deletion experiments that actually proved that the genetic code is a triplet code, so that 3 bases code for one amino acid. At that point, people started to work on trying to figure out exactly which codons called for which amino acids. So some of the first experiments that were done were making some synthetic RNAs of long strings of U's and finding out, well, what kind of protein does that code for? And so as technology developed, people learned how to make these different sequences of synthetic RNAs, and finally, about 1968, the entire genetic code was worked out. And so it was figured out what each of those 64 possible combinations of bases would code for. Importantly, people discovered that all of the codons are used. So all 64 are in use, and there are just multiple codons for some of the amino acids. The name for this is it's called a degenerate code. So it's not just a one-to-one. There's sometimes multiple codons that code for the same amino acid. The other thing I'd like to mention is that this genetic code is a universal code, and all life on Earth has this very same code. There are a couple of really minor exceptions, and these are just a couple of the codons. So mitochondria and eukaryotes actually have their own ribosomes. Just a couple of them are slightly different, but there's really the exceptions are very, very rare. So this is what was actually figured out. This table is the genetic code table, and We'll practice in class doing some translation using this table. Essentially, just look for the first base over on the left side here. So all the ones that start in U are in the first row here. All the ones that start in C are the second row. And then the second base is listed in the columns. And then the third base is found. So all of the ones that start with UU, all four of the possibilities in the third position are listed in that same box. So a couple of things I want to point out. The very first amino acid of a protein is always a methionine, and there is only one methionine codon, and that is found right here, AUG. One thing I want to mention is that although methionine is always the first amino acid, you can also have methionines in the middle of a protein. The start codon, that AUG, determines which reading frame that messenger RNA gets translated in. And then at the end, there's going to be a stop codon, and there are actually 3 different possibilities for a stop codon. They're shown here. The actual translation of nucleotide code to amino acid code is done by these really important RNAs called transfer RNAs. Now, transfer RNAs, remember, they fall into this category of a functional RNA. So that would be a gene whose final product is the RNA. When the transfer RNAs are made, they actually fold into sort of a cloverleaf shape, which looks something like this. Here is more of a space-filling structure on the left. There are a couple of really important parts of this. First of all, there's the amino acid attachment site, and this would be up at the top here. And then there's the anticodon. And the anticodon is the thing that will actually physically base pair with the codon on the messenger RNA. Anticodons are always going to pair in an antiparallel fashion with that messenger RNA. And because that is the case, this is the one exception where we're actually going to list an RNA in a 3' to 5' direction. So we want to keep that messenger RNA in the 5' to 3', so 5' on the left, 3' on the right, and so we're going to list that anticodon in the 3' to 5' left to right orientation. Just want to mention one more thing. You notice there's a couple of strange characters on this transfer RNA. These are simply bases that have received some kind of post-transcriptional modification. So there are some enzymes that bind to it and just modify them slightly. Now, transfer RNAs have to have their correct amino acid bound onto them. That amino acid will be bound onto the 3' end of that transfer RNA, and there is a whole class of proteins which are called aminoacyl tRNA synthetases, which are the proteins that will put the correct amino acid on each of those transfer RNAs. When we have that transfer RNA with the amino acid, we call that a charged transfer RNA. The last thing I'd like to address in this video are the number of transfer RNAs that are needed. And so there are 2 numbers here. There's the 64 different codons and there's 20 different amino acids. Now, it turns out that the number of different transfer RNAs that are needed is actually somewhere in between these 2 numbers. So we do definitely need a transfer RNA to bind to each and every codon. However, it turns out that some transfer RNAs can bind to multiple different codons. This is actually due to something called Wobble. Now, here is the transfer RNA, and here would be the messenger RNA. And in the first position and the second position of the codon, we have to have perfect base pairing. So A's with U's, G's with C's. However, in that third position, there is some promiscuity in what that third base can actually pair with. Now, importantly, that third base can't just pair with anything. It has to follow the Wobble rules. Now, the Wobble rules are listed here, and I don't want you to memorize these, but I do expect you to be able to interpret these rules. So, for example, if in that third position you have a G, so this would be the 5' end of the anticodon, it turns out that G can pair with a C, which would be the normal pairing partner. It can also pair with a U. If you would have a C in that third position, it can only do the normal base pairing with the G. Also, if you have an A in that third position, it can only do the normal base pairing with the U. If you have a U in the third position, however, it can pair with either an A or a G. And so if you end with either a G or a U, you have this wobble possibility. There's also one other thing that can happen. If that third base is actually post-transcriptionally modified, so it started out as an adenine and it actually got changed to a different base, which is something called an inosine. An inosine can pair with 3 different bases on the messenger RNA. So inosine could either pair with U, C, or A. And the formation of a tRNA that has an inosine in that third position, just want to mention, would be a very highly regulated process. So certain tRNAs might always get converted into inosine by an enzyme, whereas other tRNAs would never get converted into inosine. The last thing I'd like to do is just go through an example of how Wobble can be used. So we'll talk about this example, serine. So there are actually 6 codons that call for serine, and this is one of the more complicated situations for tRNAs. So there are 6 different ones. It turns out that there are only 3 required transfer RNAs, however. So this figure that we looked at is actually an example of one of the transfer RNAs for serine. If we look at the codons UCU and UCC can both be bound by this anticodon tRNA that is AGG. So remember, G can pair with either C or U. What about those other serine codons? Some other ones we have are UCA and UCG. And these 2 codons can actually be bound by the anticodon AGU. So A and G will specifically complementary base pair with the U and the C, the first 2 positions of the codons. And the U in the third position, if we look down here at our Wobble rules, a U in the third position can actually pair with either an A or a G. And so this particular anticodon, AGU, can pair with UCA, and it can also pair with UCG. Finally, there are 2 more serine codons, so AGU and AGC, and these ones can both be bound by the same anticodon, which would be UCG. Again, the first 2 pair perfectly. The third position is a G, which can pair with either a C or a U.